Hey Team Bio, welcome to your screencast on kind of reminder of chloroplast structure. It's really an introduction to photosystems and pigments, and really what we're doing in this screencast is giving you an introduction to a series of reactions in photosynthesis, which are the light-dependent reactions. Boom, boom, boom. What do I want to say? Okay, so this screencast is an intro to photosynthesis, kind of part, P-A-R-T, come on, part 1, the light-dependent reactions. The second part of photosynthesis, part 2, are the light-independent reactions. But as you might gather by the name, this part of photosynthesis is dependent on the presence of light. And then another reminder, or maybe not a reminder, but an introduction, something you may or may not know. Photosynthesis is when we take CO2 and we slam molecules of CO2 together using energy from the sun and electrons stripped from water and turn it into glucose and oxygen, molecular oxygen. So this is the overall unbalanced chemical equation for photosynthesis. But you're going to start to see how these different parts come together, how the carbon dioxide is used to make glucose, what we're doing with the oxygen, or sorry, the water, and how the oxygen is being formed. So overall, by the end of this next week and a half, you're going to have an understanding of how all the different parts come together in this equation. Okay, so just to give you a little bit of context, we have talked about the structure of a leaf and diagrammed it out. You've even looked at leaf cross-sections underneath the microscope and labeled them. Additionally, your last night's screencast was the structure of the chloroplast. So a chloroplast is an organelle within one cell within a larger organism. So again, this is one single cell, and then the organelle, the chloroplast. So we're zooming in on the chloroplast here. Here it is labeled. And this next screencast is another series of zoom in boxes. So now we are going to be looking at a single thylakoid. So to give you some context here, here is a thylakoid. Here is the lumen, the space inside, and this is, this whole thing is the TMS, or thylakoid membrane system. And then outside here is the stroma. Not the stomata or stoma, the stroma. Okay, so I'm going to be introducing you to this section of the thylakoid, which is composed of some things called photosystems and some electron transport chains. And it's important for you to know that I've only drawn it on one part of the thylakoid right here, but know that the thylakoid membrane system is covered with these photosystems and accessory electron transport chains. So if this was an accurate representation of what was going on here, these green and yellow dots would be all over the TMS, extending out throughout the whole chloroplast. Remember that the membrane is highly folded in order so we can pack more and more of these components of photosystem or components of photosynthesis together on the membrane. Okay, so what are these little green and yellow dots? Well, I will tell you. The first green dot, unfortunately, is called photosystem 2. And the second green dot unfortunately, is called photosystem 1. Photosystem 2 is first in the chain, but photosystem 1 was discovered first, which is why they are labeled so annoyingly. Okay, so we'll start with photosystem 1. Let's dive into what it actually is. Photosystem 2 and photosystem 1 are both collections of chlorophyll molecules. What I've drawn here, each of these little popsicle sticks, is a one molecule of chlorophyll, which is actually a very beautiful molecule, and if I were to get a tattoo, it probably would be chlorophyll. So chlorophylls have these little tails, and then they have rings at the top, and within the rings is a single atom of magnesium. So magnesium is a metal atom. Here is its nucleus, and then here it has some electrons. And metals, one of their characteristics is that they easily accept and then give off electrons. It's why we use them for wires, for example, for conducting electricity. And you're going to learn this again in chemistry, but electrons, when they get excited, they can jump from an orbital that's close to the nucleus to an orbital that is further away from the nucleus. How they get excited is that they absorb electromagnetic radiation in the form of light. So imagine you have a wave of light, a photon of light, comes and strikes this electron here. It's going to cause the electron to jump up an energy level. So from this lower energy state orbital, it jumps up to a higher energy state orbital and is moving around faster. This electron has two choices. It can kind of stay at this energy level, at this higher orbital, or it can re-emit the same photon of light and drop back down to its original energy state. So this is what electrons do. They absorb photons, they hop up an energy state, they re-emit the same photon, and they drop back down in their energy state. Well, why is this important? It has to do with what's happening with these chlorophyll in the photosystem. So again, this is our photosystem right here. And here is a molecule of chlorophyll. So these chlorophylls are arranged in a really particular configuration. And they're arranged such that when some light strikes this chlorophyll, this electron in our manganese atom is going to get excited, and then it's going to fall back down, and it's going to re-emit that wavelength of light. But the way that this chlorophyll molecule is oriented is such that the light is passed on to a central chlorophyll atom or molecule. And same with this one over here. So when we have a wavelength of light strike it, it passes its light energy on to this one, and so on and so forth. We have all of these wavelengths of light being captured by all of our accessory chlorophyll molecules and then passed on to our center chlorophyll. So I think you get the point. We got a bunch of light coming in, and all of our accessory chlorophyll molecules on the outside are passing their light energy on in a very specific direction towards this special, actually a pair of chlorophylls at the center of the photosystem. So it's kind of like an antenna, not an antenna, like a oh shoot, what's it called? Like a parabolic, a satellite dish. That's what I'm looking for. So a dish the way that it's shaped is that whenever something hits it, that energy is focused in at the focus point, in the center of the parabola. And the same thing is true of the photosystem. So any light energy that hits the photosystem is going to be focused on this center chlorophyll molecule. And what happens then? Well, this electron in the magnesium ion in the center of this chlorophyll, instead of just jumping up an energy state, it actually collects enough energy entirely that it is kicked out of the orbital completely. So it's outside of, it's no longer even attached to this magnesium. And it's what we call a high energy electron, or He. This stands for high energy electron. So the purpose of this photosystem was to collect light energy that hits it from anywhere in the photosystem and pass it on to a central chlorophyll molecule, which is then going to excite an electron so much because it's all this light energy that it can kick it off and create this high energy electron. So that's what's happening in photosystem number 1. This high energy electron, so here now we have an electron. Make this a little smaller. That is a high energy electron, is then passed on to an electron transport chain. And what happens here is that each of these little yellow dots symbolizes a pump. And this high energy electron has energy that can be spent to do something. And what the energy is spent to do is to take hydrogen ions, which you know are charged and cannot flow through a membrane because of their charge, because they're hydrophilic and the membrane interior is hydrophobic. And these little yellow dots serve as pumps, which take the hydrogen and pump it into the interior of the lumen, where there's actually a much higher concentration of hydrogen ions within the lumen of the chloroplast. So it takes energy, this is active transport, to pump the hydrogen across the membrane. And where the energy is coming from is this electron. As this electron travels, is passed from pump to pump, and hydrogen is pumped through, this electron loses energy. So by the time it gets to this last part of the chain, it is now a medium energy electron, a Me, because some of its energy has been used to pump hydrogen through the membrane against its concentration gradient. So what happens here is that this medium energy electron is then passed on to this photosystem number 1, which is actually second in the chain, super annoying, and it is re-excited by light energy, and it kicks out another high energy electron, which is then passed down an electron transport chain. So I'm going to say these two, both these yellow dot things symbolize electron transport chains or systems. So you'll hear these called electron transport chains. You'll also hear them called electron transport systems. At the end of the chain, there is a special enzyme called NADP plus reductase. And NADP plus reductase takes NADP plus and a free hydrogen atom, and it picks up 2 electrons. Oops. And forms what's called NADPH. And this is carrying 2 high energy electrons. So in the first electron transport system, we use the energy of the high energy electron to pump hydrogen into the membrane. And the second electron transport system, we passed off 2 electrons to this high energy electron carrier. It's like a suitcase to carry these high energy electrons because they're high energy and they can cause damage if they're not properly transported. So we carry it in this molecule, and this molecule is going to go off to what's called the light independent reactions. Okay, the last thing I need to tell you that is important, and I realize this is turning into a long screencast, is that we're actually going to do something with this concentration gradient. Remember before March Intensive, I was telling you about how a chemiosmotic gradient, when you have a high concentration of an ion on one side of the membrane compared to another, is just like a hydroelectric dam. There's potential energy here in this concentration gradient. And if you run these ions down their concentration gradient, you can capture that energy in another form. And that's exactly what happens here. We have an enzyme. It's called ATP synthase. And ATP synthase takes the energy from hydrogen flowing down its concentration gradient and uses it to form ATP from ADP, adenosine diphosphate, and free phosphate. So, this is powered by the hydrogen concentration gradient that is formed by using the energy from this high energy electron. And so this thing, ATP, and this NADPH are both headed to help power the light independent reactions. So the 2 products of the light-dependent reactions, ATP and NADPH, are used to power the next phases of the cycle. Okay, the very last thing I'm going to tell you, and I realize this is turning into a very, very long screencast, and I apologize sincerely, is that this electron, when it's passed down the chain and eventually dropped off at NADPH, means that we've lost an electron from this original magnesium atom here. This is down an electron. So we need to supply a new electron that we're going to excite into the high energy electron. And where does that electron come from? Well, it's actually, stripped from water. We take water at the very beginning of the reaction, we add it to the reaction, and we strip an electron. So it's water that is providing that electron. This process of taking the water and stripping an electron from it is called hydrolysis, the breaking of water, the lysis of water. When we break water apart, we create H+, and we also create oxygen. So, initially, at the very beginning of photosynthesis, water is broken, an electron is stripped away. That electron is going to be turned into the high energy electron using the channeling of energy of the photosystems, and the waste product of this breakdown of water is hydrogen, which is going to contribute to our concentration gradient inside of the lumen of the chloroplast, and oxygen, which, as you know, is going to build up inside of the leaf and then leave through the stoma, or the multiple stomata. Okay, that is all. This is a little bit complicated. Don't worry. We're going to spend a lot of time working through all these different steps and talk about this over and over and over again, so you're really going to get it. Okay, see you in class.